the work I'm going to present today is mainly a modeling perspective, okay, on how we can model different mechanical systems. And to be honest, I'm going to cover quite many different, a priori, completely different topics, but in the end, I want to kind of merge everything, right? And explain you more or less my ongoing work and what I want to do for these following ongoing years. So as I said, I have decided to split this presentation in two main blocks. So first, I would like to talk about some mechanics in biological processes. So I will talk a little bit first on how we can simulate collective cell migration, some insights from a mechanistic point of view. Then some briefly, just introduce some previous work I did on brain mechanics, and then move to a priori completely different topics. So especially how we can model and how we can understand magneto-active hydrogels. And then just I really would like to discuss together on how we can use these kind of smart materials to somehow interplay with all these biological processes. So let me start with this first part. Okay, so when we look at biology, okay, we can find many different biomechanical processes, such as cell migration or traumatic brain injury. So I will give more details on these two ones, particularly later. And also in bone healing or even in cardiac function, right? When we have this kind of electric response, that is intrinsically coupled to all this mechanical pumping of the heart. So something that is clear at this stage is that all these different biological processes, in all of them, we have a very important role of mechanics. And when we look inside these mechanics, right, we find that most of the biological experimental approaches or some of the modeling techniques, just assuming this is quite simple with some just linear elastic models following Jones modulus or even linear viscoelastic approaches. But if we look inside in dependencies of these mechanics, we realize that this is quite complex, right? So we have many different dependencies, such as temperature modulating the stiffness of the biological tissue. Also, that these biological tissues are not like elastic solids. So we have some viscous contributions. And as a consequence of that, we know that we have a strain rate dependency. So the faster we apply the mechanical loading, the stiffer response the material provides. And also intrinsically linked to that, we have some viscous relaxation phenomena. So in the end, if we apply load faster enough, right, and we keep the load, we can observe with time some stress relaxation. So on top of these dependencies, we have many others. So just to mention one another, we can find some tissues like muscles or tendons given an isotropic response. So basically, it's telling us that our material is responding differently if we apply the loading in, along the fiber direction or if we apply that in the transverse way. And on top of that, a common characteristic of all these dependencies is that this is full of nonlinearity, right? So as I said before, even from not only from a modeling perspective, but also when we analyze this kind of experimental data, we need to account for these changes. So as we are reaching these large deformations, we need to account for all these nonlinearities. So just want to present today some works already published actually. The first one, I'm going to focus on collective cell migration, and then I'm going to move to just quite briefly on how to model, right, and the implication of traumatic brain injury. So regarding this work in collective cell migration, so to be honest, this is quite a new topic for me that I started with some people here in my university. So this work was published this year in Stream Mechanics Letters with Arrate Munio Barrutia. And well, so basically the idea here is that when we look at one cell, we realize that there are some protrusion, some contraction, some expansion. As this is far from being an isolated system, we know there are many different interaction forces with the surrounding extracellular media, the substrate, and also as soon as this is somehow connected to other cells, we have also some interaction forces with them. So in the end, all these mechanical forces plays a given role. And as soon as we have all the different cells other each other, we can have some transmission of these mechanical forces. And in the end, well, something that I think is quite interesting here is this collective cell migration response. So just to provide you a couple of examples, I have this picture here in cancer cell invasion. So I have to thank Professor Dennis Beard from Johns Hopkins University, really, because I think this is quite clear how in this cancer cell invasion we can see kind of continuum-like migration response. And also here for wound healing, epithelial wound healing. So this is courtesy of Professor Xavier Trepat from Barcelona. So many thanks to him, really. Because in this video, we can really see all these different mechanics interplaying together. So if we stop the video here, we can see this kind of protrusion. So in the end, we have some mechanical forces at the individual cell level, and we can see how all these forces somehow are transmitted to the whole continuum. So we have this kind of continuum-like migration. And at this point, I really want to highlight that what we are observing here is not the small deformation. So for sure, we're going to deal with some large deformation and non-linearities. So let me go to this process. So from a more theoretical perspective, let's analyze the different governing equations we can find here. So I decided to reduce to these three ones. Actually, we'll focus on the latter two ones. But we can imagine, and actually this is some ongoing work we're already doing, that we can have on top of that any kind of proliferation dynamics or other chemical-like processes we can couple to mechanic systems. So regarding the concentration of the contractile actomyosin, so basically we can follow this actomyosin concentration and we can link that to a kind of active stress. So it's going to determine the dynamics of the cell contracting or expanding. And well, so it's important to think about it, that we have kind of perfectly spherical cell. In the end, all these forces somehow are going to be canceled out. So this is important to jump to this second one. So this is cell polarization dynamics. It means, okay, so our cells can change in shape and orientation. And in the end, that way, all these contractions or these forces are going to be somehow directed. And then let me introduce this third one that just my personal perspective, this is the most important one for me at least. This is mechanical balance. So this is kind of taking a cocktail and put everything together, right? So in the end, we're going to account for all these different forces and all different interactions between different cells in a global or continuum manner. And in the end, we're going to get quite of the resultant forces acting on the system and how this is going to determine the fate of this collective migration. So keeping this in mind, we wanted to focus here on a very specific problem. So we took actually from Trepat's group in this paper of Sanjay et al. In Science. So basically here, this is experimental data on some epithelial monolayers, okay? So a given bands of cells connected to each other that we can put on top of different substrates. So what we can observe here is that in this left-hand side image, we have all this system, okay, on a uniform substrate. So what we can see is that we start from this kind of light area, and we can see how this whole system is expanding. So it's migrating in a quite symmetric fashion. So then we can put exactly the same system, well, exactly in the same manner, epithelial monolayer on top of a heterogeneous substrate. So in this case, what we're going to have here is kind of a stiffness gradient through this substrate. So what we can observe is that all this collective system is moving toward that stiffer region. So then we wanted to look into the published literature, right? So how we can explain that? And in the end, most of the experimental approaches and actually some nice modeling works are suggesting the following. So we have this kind of cellular system that somehow is linked to the substrate by different focal adhesions. So if we have a stiffer gradient, so different stiffness of the substrate locally, we can expect that the forces here in the stiffer region are larger than in the softer one. So in the end, we have kind of resultant force acting on the system and directing this whole process. So we look further in the literature and we can find some experimental evidences to say, okay, so a priori, this is not only the propulsion force, so we can have other important dependencies, right? So regarding this polarization dynamics that, as we can see here, is going to be widely linked to how these forces are exerted, we discovered that this is quite influenced by the magnitude of the substrate stiffness and also by different stiffness gradients. So cells tend to align to that stiffer regions. So at this stage, we decided to put all these physics together and try to develop a model to see which are the, let's say, the consequences of all these hypotheses and how we can use these models to complement some experiments and get a further insights into this process. So I have reduced quite a lot the mathematical formulation for this problem. And well, so let's start with the mechanical balance in the uniform configuration here. So we will have kind of put together these internal and external forces. So here H is the height of the epithelial monolayer, and we have the gradient of the Cauchy stress tensor. And then we are going to have some traction forces. So these traction forces are going to determine how this cell continuum is going to deform. So we can see here that we will have any kind of propulsion force, okay, with a magnitude that a priori and following these previous approaches can depend or will depend on the stiffness substrate. And then this cellular polarization is going to determine somehow the orientation of that traction force. And then, of course, as soon as we have any given system moving on top of another, we're going to get some frictional forces determined by this friction coefficient and the velocity of the system. So once we define that, we want to define also what is happening internally, right? So how is the evolution of this mechanical stress within the cellular system? And to do so, we made use of some hyperelastic theories. And well, just to mention here that although we can include many other dependencies, we wanted to reduce the system at this stage to some simple model. So just using this Neo-Hookean model, right, for incompressibility conditions. And so we define this mechanical potential, that this is kind of energy stored within the system. And then using, well, solid mechanics theory, we can consistently derive in order to get the relationship between deformation state of the cellular system given by deformation gradient and our Cauchy stress tensor. So just to mention here that we are assuming this response, this deformation as an incompressible system. So we have this first volumetric component where P is the pressure that we're going to get actually in the implementation as an extra degree of freedom. So this is going to be given by consistency of this incompressibility condition and a penalty factor. And then this mu cell that is describing the shear modulus of the cell. So just to mention that here, we are measuring not only the intrinsic mechanical properties of the cell, but also somehow accounting for this adhesion strength between the different ones. So let me come back here. So when we're looking at the traction forces, we see that it depends on the propulsion force, but somehow this is directed by the polarization of the cell. So our point is, okay, so as the whole system is moving, we can expect that this polarization is going to change during the process. So in order to define that, we're aware we can, actually we included here many other dependencies that are demonstrated from experiments, like a dependence with velocity and others, but we wanted to reduce to that simple dynamics for the cell polarization in order to see clearly and isolate the different contributions of the hypothesis I mentioned before. So first, this blue term is saying, okay, so because of this substrate stiffness gradient, we can have some cells that polarize to one direction, right? So then, as soon as we have all this system, so we have a kind of continuum system, these other cells are going to experience kind of diffusion-like process, so that they're going to trend to align to that direction. So then, even if we have a constant propulsion force, what we can observe is that our resultant force is going to guide all this system to move towards the stiffer region. So once we have that here, so actually I skip here the whole implementation details. We implemented that in a finite element code and integrated that in with implicit integration algorithms. And once we had everything implemented, we wanted to test the model, right? So at this stage, it's important to say that we didn't feed the parameters. So we got all these important parameters directly from physical meaning or from experimental data actually provided by these collaborators. And basically, the first thing we did is test the model on top of homogeneous substrate. So we took this experimental data I mentioned before from Sanger et al. In Science, and what we can observe here is these black dots are the experimental results, and these red lines are our model prediction. So by means of migration dynamics, what we are observing here is distance to midline. So it's like kind of the expansion on the epithelial monolayer a long time, up to 10 hours. And well, so according to the results, we found a very good agreement between model prediction and experimental data. So we're quite happy with that, but wanted somehow an extra validation, a double check of the model predictions. So in that terms, we wanted to look by means of mechanics. So actually, for these same simulations, we look into the stress tractions within the material. So the stress times this layer height. And what we can observe is a really good agreement between experimental data and modeling results. And actually, if we look at here in this graph, we can observe that this maximum stress traction in this dashed black line is actually almost exactly the same than the one predicted by the model. So we're happy with that, and we move to the following set of simulations. So let's see now how we can model this kind of direct immigration towards the stiffer regions. So to this end, we decided to split these simulations to isolate the two different hypotheses in two different sets. So all the simulations in red are to analyze stiffness-dependent polarization. So at this point, we're saying, okay, so my propulsion force is going to remain constant, and now I'm going to get this dependence with substitute stiffness just in the polarization. And then for the blue simulations, we have in the other way around. So we are considering no effect of the substrate on the polarization, and all this dependence, okay, it's coming here into the propulsion force magnitude. So if we look at these migration dynamics, actually, a priori, we can say, okay, so both hypotheses are providing us an explanation to explain that response, that durotaxic response. And actually, we can say it's pretty well for both of them, and we get the dynamics. But then if we move to mechanics, from a mechanics view, let me go there here because I think it's clear. So we can see that for this stiffness-dependent polarization, we can explain this given movement towards the stiffer region, and we can keep this symmetry in the stress distribution, and also we only need, or it only means or implies a slightly increase in intercellular stress. But if we follow up this kind of propulsion force dependent approaches, what we can see is that we are breaking the symmetry of the stress concentration, and actually, if we want to explain it by that, it means that we are assuming a higher increase in stiffness, in intercellular stress. So I wanted to highlight here how we can use this kind of modeling approaches, okay, accounting for all these mechanics, and use these models to support experiments and test hypotheses. So it's not only that we provide, we analyze a problem from an experimental point of view or a modeling point of view, but let's try to use all these different methodologies, right? And test each problem from different perspective. And also, one highlight I wanted to point out here is that this biological process is clear that it's not only about the mechanics within the cell, but also all these processes depends a lot on these surrounding mechanics, on this extracellular matrix or environment. So I want to briefly introduce now some previous work I did in what I call brief story in brain mechanics, just to provide other examples of these mechanical dependencies on biological processes. So actually, so this is some work I did during my postdoc in Oxford. So you have here like the two different papers already published in the group of Professor Untitled Chapter, where basically we were focused on understanding central nervous system injuries. So the idea here is, okay, so when we have an impact on our head, could be sport-related activities, fall of a person, or even here in kind of military context, when we have a blast explosion, or we have this mass of air impact in our head. We know there are some consequences, right? I mean, of pathology, brain dysfunction. And actually, we know that there are some biomarkers, right, like oxidative stress that can tell us, okay, so you are damaged or not. To us, it was clear that, okay, so if this is happening, so the input is a mechanical input. So somehow it implies that our brain tissue or even the spinal cord tissue is somehow mechanically deformed. So then we should be able to get some mechanical variables that govern all these injuries, right? So our idea was, okay, so let's try to get some mechanical threshold that this is telling us, okay, so from this threshold you can have any kind of dysfunction, and if you don't reach that, we can see if this is healthy or not. So not only looking at this kind of overall consequences, but also trying to relate that across different scales. So actually what we know is that if we go to the cellular level or tissue level, actually all these brain dysfunction, etc., are quite related to the propagation of these electric signals along the central nervous system. And there is evidence of some mechanical works that if we introduce some mechanical deformation, we are somehow damaging or altering this kind of action potential, so this kind of electrical signal. So in order to study that, well, we did this in collaboration. So actually all the experimental work is done in Purdue University with Professor Reggie. And basically these guys developed some animal models, okay? So we were able to impose this kind of blast explosion on a rat model. And inside the brain, we put, well, actually on top of the brain, we put chunks of magnets. So they were able, right, to track all the displacement of the brain during this blast explosion. And after that, it can provide all the different damage regions by means of some biomarker like oxidative stress here, and also get any kind of overlap, complementing that with cognitive deficit tests. So let's try if the rat is working well by means of auditory system, olfactory system, or others. And then we actually got really good overlap between this cognitive deficit test and this biomarker distribution. So the idea I want to mention here is that, okay, so from an experimental point of view, we can go ahead, but at some point, if we want to look into the mechanics and we want to look what is happening during this whole event, we need somehow to complement with some computational approaches. So in this regard, we developed some finite element models representing different regions of the head of the rat, different tissues. We put all different constitutive equations accounting for all these strain rate dependencies, non-linearities, etc. And we were able to get very good validation by means of this displacement and some other markers. And then we were able to see which mechanical variables we can use in order to explain this kind of threshold that is leading to damaged regions or not. So overall, this is quite phenomenologically approached, but it's providing us some threshold, right? So in the end, it was kind of a strain energy rate that we discovered. So also what these models allow us is, okay, so now let's take part of the brain region, right? And develop some multi-physics approach. So I actually don't have time to explain all of this, but I'm really happy to discuss it together. So we can reproduce all these electrophysiological signal or this electrophysiological response. So we can see how these electric signals propagating along the axon at the cellular level or tissue level, taking into account the direction of the axons, and we can see how these mechanics are interplaying with these effects. So other things that all these computational models allow us is, okay, so let's jump from rat to a human. So obviously we cannot carry out this kind of experiments on humans. And what we did is reconstruct kind of finite element model, right? That this is not only the geometry, but we can also add some internal variables to every single element. So in this case, we were able to track different axonal orientations and get for every single element in the white matter, the axonal orientation and how they are dispersed, right? How, if they are well aligned or not. And then we test some experiment numerical blast scenarios and discover that, okay, we can get with this approach, with threshold, that the damaged regions are going to be somewhere here. So with some neurologists, we analyze that this is quite related to deficits in learning and memory, emotional memories, or even sensory motor systems. So we got a priori good, let's say, agreement with some deficit we can observe in this kind of military people. So just wanted to present that to provide an overall view of how mechanics can affect these different biological processes and how we can develop computational models to help with experiments. So now, well, so trust me, I'm going to jump to a completely different topic, but trust me that in the end, somehow I want to merge both of them and well, let's discuss together about it. So let me introduce now magneto-active polymers. So I'm aware, so is Costas and other people in your lab. So actually, in this regard, you have much more experience than me playing with magneto-active polymers. So just to conceptualize that, this kind of smart composite, right? That this is composed by a polymeric matrix, and we can fill that with some magnetic particles. So that as soon as we apply any kind of external magnetic field, we're going to generate some interaction forces between the particles, and then what we're going to get is that by mechanical balance, it's going to introduce some deformation, some changes in the mechanical properties of the overall composites, or even some mechanical deformations. And if we look at this kind of scheme, this kind of cartoon, we can realize that we're going to have different magneto-active polymers in nature. So depending on the polymeric matrix, so we have from thermoplastic polymers to elastomers or even hydrogels, okay? So it's going to determine more or less this magnetorheological effect and the stiffness of our material, but also we can change the different magnetic particles. And regarding the latter, we can split it into two main groups. So one is hard magnetic particles that basically is kind of particles that present a given magnetization, so a given residual magnetic flux density vector. And in this regard, it allows us to somehow anticipate how this system is going to react, is going to respond to external magnetic fields. And let's try to program some safe chains of actuated robotics or in biomedicine. So just wanted to highlight this paper by Professor Sao, published in Nature, where they present quite, I think, pioneering 3D printing technique to program this kind of smart materials. And actually, they published this work in JMPS with more or less some basis to address this problem. I know now, for example, so Costas is also working on that and advancing in this field, right? So the other kind of magnetic particles we can find, so magnetics, right? So basically, this is particles appear without any given residual magnetization vector. And then as soon as we apply this external field, we are introducing some changes in this magnetization, and we get the different responses. So just wanted to focus on these latter ones. And when we link here, so we see different, let's say, polymeric matrices we can use. So this is usually is common to find elastomers as the polymeric matrix. But here I wanted to, so this is the first link to the previous part of the presentation. If you realize, we can use hydrogels that cover this stiffness range, where we can find different skins, muscles, brain, spinal cord tissues, cells, etc. So it opens the possibility to interact and have an interplay between this smart system and the biological tissues. And well, of course, I'm going to be the first one doing it, so there's already some published work on that, as we can find here for soft actuators, nanorobotics, or even drug delivery system, right? So I think this is quite interesting, as we can take this kind of small particles of magneto-active hydrogels, and we can put inside some drug. So making use of this external magnetic field, we can direct these particles, and then once located in the desired region, we can apply this kind of magnetic field in order to introduce some mechanical or even thermal changes. So we get this drug delivered there. So when looking at this problem, what we realize is that this is a little bit more complex even, because now this is not only mechanics and magnetics, but by the nature of these hydrogels, we're going to have some solvent diffusion processes happening there. So this is actually what I want to present now, that actually is a very recent paper we chat with Professor Landis from Austin. So basically wanted to provide kind of general constitutive and finite element framework to analyze all these interplays between magnetics, mechanics, and solvent diffusion, and look these interplays, these couplings, along different time scales. So just to start with that, well, the three problems we have and the governing equations here expressed in reference configuration for both volume and on surface. Here, the mechanical balance, where we have here the gradient of the first Payola-Kirchhoff stress tensor, we have F and T sub 0, these external forces, and then the normal unit vector to surface. Then for the magnetic problem, we can have in a very similar fashion. So these two different magnetostatic equations. The first one for the magnetic flux density vector, the second one for the magnetic field. And then for the diffusion process, what we need to define is this is the solvent concentration per unit reference volume. So basically what we are saying here is that we're going to have a local evolution of the solvent concentration that is going to depend on these RS and IS being kind of external sources of, so basically how the number of solvent molecules were injected into the system, here per unit volume, here per unit surface. And then this JS is the solvent flux. So this is a vector that basically is determining how all these molecules, solvent molecules, are moving within our system. So just to have more abstract and let's say a theoretical point of view of which are the potential interplays between different processes, so it's clear in the magnetomechanical problem that as soon as we apply any given external magnetic field, we're introducing some interaction forces between the particles, and then to satisfy this given mechanical balance, and depending on the boundary conditions, we're going to have some deformations of the polymeric composite. And well, if we think about it, so at this stage, if we start now to deform the material, we can also change the nature of these kind of electromagnetic forces and even change the nature from attraction to repulsion. And when we look at these mechanics interplays or coupling with solvent diffusion, we can think about kind of response, right? So we have this polymeric network with the different solvent molecules, and when we deform it fast, right, we get a short range motion, right? So here actually what we are creating is kind of a gradients in the chemical potential of this solvent, and also some mechanical gradients by means of pressure or other mechanical variables. So in the end, if we keep it for the long range motion, what we're going to see is that the system tries to come back to equilibrium, right? So try to suppress this kind of solvent chemical potential gradients. So in the end, we're going to have some new molecules coming here inside the polymeric network and leading to swelling shrinkage. So in the end, obviously, we have a strong coupling with mechanics. And just to mention that as this, there is a link here between magnetics and mechanics, and there is a link here between solvent diffusion and mechanics. Obviously, indirectly, we also have kind of interplay between magnetics and solvent diffusion. So having this in mind, we try to put everything together. So we made use of non-equilibrium thermodynamics theory. So just to mention first, so we describe here kind of a total free energy in two different components. So this first one is the free energy associated to the material, and then this second one is kind of the free space associated energy. So just to mention here that if you realize we have included the viscous contribution to the deformation gradient as independent variable. So basically we made use of the split multiplicative decomposition of the deformation gradient into an elastic and a viscous part. And this is done in order to be able to account for well, strain rate dependency, viscous relaxation, and others. So basically this viscoelastic nature of the polymeric network. So playing with this definition of the free energy, and also playing with taking into account different governing equations I mentioned before, we put everything together here in this kind of inequality for the free energy rate. And then directly from here, applying some Coleman and Knoll arguments, we can get the following constitutive equations. So just mentioned, we also made use of the Legendre transform, just for the sake of convenience, to have the magnetic field and the chemical potential as independent variables, just to help us with implementation. So in the end, we get constitutive relations for deformation gradient, Viola-Kirchhoff stress tensor, also for the magnetic variables, and also for the solvent concentration and this solvent chemical potential, this μs. And then these last two terms actually are going to provide this kind of consistency conditions to define the kinetic law. So the solvent flux, that we took this expression where this ds is kind of mobility tensor, symmetric and positive definite, and then we have this gradient of the solvent, a chemical potential. And there's the second one is going to determine somehow which kind of evolution for the viscous flow rule we can impose. So at this stage, I really wanted to introduce what we did here. So a priori, it can be, it seems that this is not, let's say, efficiency from a computational point of view at all. So let's say that in order to solve this problem in a given implicit framework, we just need the displacement vector u, okay? So from this, we can define deformation tensor, a magnetic scalar potential. To define this kind of magnetic field, we can also use other approaches based on vectorial independent variable. And for the solvent diffusion process, the chemical potential, right? But in this case, our point of view was the following. So actually we want to have something general, right? So now we're doing, and we're going to do more experiments for this kind of hydrogel magnetic systems. And in the end, we don't really know for every single material which kind of tendencies, which kind of dependencies we're going to have, right? So we wanted something general that allow us to quickly and easily test different combinations of all these different, constitutive equations. So we can test different hyperelastic models for the mechanics, different magnetic contributions to the stress state, also different viscous flow rules, right? So what we did to do so is using Phoenix, that this is an open source finite element code, okay? So basically it allows for defining manually different parts of these stiffness matrices, or we can just use kind of automatic numerical differentiation, right? So numerical computation of all these stiffness matrices by defining just some residuals and some independent variables. So in that sense, our idea was, okay, so let's introduce the increment of this viscous part deformation gradient as an internal variable, so as an extra degree of freedom. And then let's put it together within a kind of residual that is imposed in the general form of the viscous flow rule. So basically, it's determining how these evolve consistently with this viscous contribution to the rate of deformation tensor. So basically, by imposing the residual, we're going to get somehow, we can define any given different viscous flow rule. And then we can put it into Phoenix. We can test many different combinations of constitutive equations, constitutive fields for every single bit, and we can easily get a first approach. So, of course, this is not efficient from a computational point of view, but we can test many others, and once we have our optimal solution, go for defining these matrices manually. So once we had that together, and let me present it more fast now. So we decided, okay, let's try our system. So we try actually different constitutive relationships, just to mention the one we included in the paper. So we decided to split this free energy into a polymeric network component. So basically we follow this flow-to-Renner approach, where we have this first contribution, this is kind of purely hyperelastic part, and then the second part related to just the elastic contribution of deformation gradient, and it allows us to introduce kind of viscous dependency, right? So then a magnetic component. So in this case, we say, okay, so let's try just with the most simple approach, so kind of linear model. And then the last term, that this is related to polymer solvent mixing components. So basically, we're seeing here the energy potential associated to this interaction between the polymeric network and the solvent. And just to complete the framework, we impose this volumetric constraint. So we are saying, okay, so our polymeric network can be assumed as incompressible, and then the changes in volume are going to be associated to how is this volume associated to the solvent inside. So this concentration of the solvent times the volume of this solvent molecule. So we can see that depending on the solvent concentration, we're going to determine kind of the volumetric response. So once we have that, just we can directly apply different constitutive equations in order to get the stress, the magnetic field, and the solvent concentration. Then we define consistent mobility tensor and viscous flow rules. So for this one, we follow this approach with this kind of solvent diffusivity. And for the viscous flow rule, something simple as well, defining here a viscosity parameter. So, let me show you some dynamics we got. So first of all, I'm sorry because I didn't realize I included here one example with a coarse mesh, okay? But actually we can see all the different dynamics in the process. So the idea here is that what we are plotting here is the solvent concentration. So let's say we take one initial Magneto-active hydrogel with a given concentration, and we put it into a given solvent, right, media. So the first thing we're going to see is how we are reducing this kind of chemical potential gradient and pressure gradient so that we have a kind of diffusion process until we get stabilization and equilibrium of our system. So once we get that, we decided, okay, let's see how this is going to respond to our external magnetic field. So what we can see is that we have quite a fast response, right? When we can observe some compression or some mechanical deformation of the system. So here, this is interesting to observe that this is not homogeneous deformation. So basically, let's think about it. So these corner regions are actually in full contact with the solvent. So actually here, we have this kind of chemical potential. But in the internal regions, we're going to get trapped some solvent. So then we have, let's say, instantaneous response here. And then if we keep it into the long time range, what we're going to serve is kind of transient response in order to get my new equilibrium state according to the external magnetic conditions I have imposed. So just to analyze this a little bit more, I decided to include here some analysis we performed. So the first one is here, looking at the solvent diffusion effect. So here in red, it's like kind of assuming we're going to have a given coating on the hydrogel. So we don't allow for solvent diffusion. So what we can see is that we start for a given stretch, okay, initial state of the hydrogel with some concentration of solvent inside. So as we apply here the magnetic field, we can see a given deformation, and then we just get the equilibrium state. So there is no changes with time. And if we realize here, well, it's not clear, but I'm plotting, well, two different stretches, longitudinal stretches in Y for two different points in the hydrogel. So we get a homogeneous response and always in equilibrium, right, without any kind of transient response. But if we allow now for solvent diffusion, so this is the video I presented before, we can see it in black. So these black dots are kind of the corner, okay? The stretch stay in the corner. So we can see that we get a given mechanical deformation, and actually this is kept a long time. But what is happening somewhere in the middle, so somewhere on top of the face, right, that this is not fully surrounded by the solvent, is that we get an instantaneous response, and then this deformation is going to continue in kind of transient response, okay, due to this solvent diffusion until reaching this homogeneous state within the solvent. So regarding that, we wanted to analyze now, okay, so what is going to happen if instead of applying here this magnetic field as idealized and homogeneous within the whole system, let's try to apply that from the surrounding, right? So what we can see here is that we get a very similar response than here in black. We have this corner getting the equilibrium stretch, and then we have kind of a transient response for the middle. And what we can see here is that even in the final long-term range, we don't have an homogeneous deformation. And basically here, we wanted to highlight that in the end, with this kind of soft hydrogels, magneto-active hydrogels, we apply this kind of magnetic field on the surrounding, but in the end, we're going to experience some fringing magnetic effects. So basically, because of the shape of the geometry of the hydrogel, as well as the nature of the magnetic permeability of the different particles inside, we can get different distribution of this magnetic field, especially higher in the boundary. So we're going to have some heterogeneous deformation in the end. And just to conclude this analysis, now we wanted to see, okay, so this is kind of long time range. So let's zoom this region where we directly apply the magnetic field and see how are the dynamics here. So to do so, we remove, in order to isolate this effect, we remove this solvent diffusion, right? So no solvent diffusion is allowed here. And what we can see is that we also have some transient response, right? And in this case, what we are doing is, until this vertical red line, we are increasing the magnetic field, and then we keep it constant. And we realize that because of this viscoelastic nature of the polymeric network, what we have is that depending on the rate of this magnetic field application, we have a different stiffness, so reaching the mechanical balance equilibrium for a different deformation state. And then what we can observe is, for this short time response, we're going to have any kind of viscous relaxation, that actually this is something also we also observe in biological tissues. So I want to highlight here, so we have many different responses playing at the same time in different physical processes. And actually, I would like to conclude this presentation with the following. So something we realized is that, okay, so we realized that there are many different biological processes where mechanics plays an important role, right? So when we look at these different stiffness range, we realize, okay, so maybe using hydrogels, we are playing the same system. So by using these hydrogel-based systems, we can have a good interaction by means of stiffness mimicking the mechanical properties of the biological tissues. So the idea here is, okay, so let's try to control the cellular mechanical environment. Let's try to control that by applying remote magnetic fields. So the idea here is that, okay, so from an experimental point of view, to be honest, I don't think this is approachable because there are too many different things happening here. So understanding what is going on is super difficult, not only because of the smart material, but also because of all these dependencies in the different biological tissues, right? Through this biomechanical process. So they propose is, let's try to combine experiments, theory and modeling. Let's try to have given theoretical and computational framework so that we can anticipate what is going to happen in my substrate. So I can anticipate how my biological tissue is going to respond, and let's try to anticipate these interactions between the substrate and the cellular system, and let's try to influence somehow in a remote manner on these biological processes. So this is actually some ongoing work that we are doing, and I hope to do in the following years. I actually got the ARC that Estela mentioned before, to go into this direction. So we are getting some nice preliminary results. So I'm very happy, and I have positive hopes here. But well, so this is just preliminary work, and looking at your experience, especially in all the Magneto-active system. So really happy to get any comment, feedback, room for collaboration, or even if you have critics to the work, really happy to discuss it together now.